Okay, so welcome to this next video in which we are discussing the intracellular domain of the Cys-Loop ligand-gated ion channels. Okay, so before I go any further, I just want to stress something that I feel like I should have stressed right at the beginning, which is that all Cys-Loop ligand-gated ion channels have this intracellular domain. We are studying it for all of these at the same time, basically. We're not narrowing our vision down to just one. And I also want to say that in my previous videos in this playlist where we've been studying the pore, for instance, of the nicotinic acetylcholine receptor, the principles that we've seen there will carry on across all of these. So, for instance, the fact that the M2 membrane-spanning alpha helix is the one that lines the pore, that's true in all of them. Okay, yes, you won't have rings of negative charge for the GABA-A and glycine receptors the way you have for the nicotinic acetylcholine receptor, but because they conduct negative ions, so you wouldn't want rings of negative charge. But the 5-HT3 receptors, which do conduct positive charge, they will also have these negative rings. So basically, the Cys-Loop ligand-gated ion channel family, their structures are extremely similar, and certainly with regards to the intracellular domain, we can study them all together, basically. Okay, right. So now what we're going to do is do an experiment concerning these 5-HT3 receptors, which tells us something interesting about the role of these intracellular loops in controlling the conductance of ions through the channel. Okay, right. So let me tell you something about the 5-HT3 receptors. So, the 5-HT3 receptors then, their endogenous ligand is 5-HT or serotonin. So these are the 5-HT3 receptors. Okay, so their endogenous ligand is serotonin, or 5-hydroxytryptamine. Now, most receptors that we have in the brain for serotonin or 5-hydroxytryptamine are G-protein coupled receptors. So this 5-HT3 family contains all the 5-HT receptors that are known, which are ligand-gated ion channels. Okay? Right, so let me now discuss their structure. So we know that they are a Cys-Loop ligand-gated ion channel. So they have these 5 subunits joined together in a pentamer. Now the question is, how many genes do we have for subunits of 5-HT3 receptors? Okay, well the answer is we have 5 genes, but only 2 of these have been characterized at all well. So we have the 5-HT3A gene, okay? We have the 5-HT3B gene, okay? We have the 5-HT3C gene, okay? Then it keeps going on, 5-HT3D, and I might as well finish this now. And then finally, 5-HT3E. So we have these 5 genes in the human genome, which have all got slightly different sequences of amino acids and all got slightly different sequences of organic bases, which leads to a protein which has a slightly different sequence of amino acids. However, the overall protein that you produce from all 5 of these genes is similar enough to do a similar function, and all of them are counted as subunits of the 5-HT3 receptor. And I do want to stress, we've been talking about Cys-Loop ligand-gated ion channels in general, but basically you do not get chimeras. You do not get 5-HT3 receptor subunits mixing with nicotinic acetylcholine receptor subunits. So we've been talking about how similar they are, but basically the subunits of each of those 4 types of Cys-Loop ligand-gated ion channel that I've shown you do not mix. Basically, you have subunits for the nicotinic acetylcholine receptor, you have subunits that can make up 5-HT3 receptors, shown here, you have subunits for the GABA-A receptors, and you have subunits for the glycine receptors. And when you assemble a pentamer, you can't mix and match between those families, basically. Okay, so you don't get any sort of crazy chimeras. However, in a family, so in the family of 5-HT3 receptors, you can build, you can mix and match, basically. You can build pentamers which have different subunits in, not all of the receptor subunits in all 5 positions are the same one. Okay, so there aren't actually many of these known. So one that you can make, so if we're looking now at the top of our 5-HT3 receptor, so here's the pore, here are these 5 protein subunits that make it up. One that you can make is basically what's known as the 5-HT3A Homo Pentamer. Okay, and that is exactly what it says on the tin. It is a 5-HT3 receptor that you make by putting 5 5-HT3A proteins together. Okay, so in each one of these slots you are going to put a 5-HT3A protein. Okay, so let me continue this on now. So each one of them is a 5-HT3A protein. So it's a Homo Pentamer. You go to the 5-HT3A gene, you use that gene 5 times to make 5 identical proteins, you put them all, stick them all together, and you've got your 5-HT3A Homo Pentamer. Okay, that's one that we've seen physiologically. The other one is, if I show this here, is the 5-HT3AB Hetero Pentamer. So let me put these in here. So, now, I cannot show you the exact stoichiometry of the 5-HT3A and the 5-HT3B subunits. I'd love to show you that, but it's just not known. So what we do know is that there exist channels, okay, which are 5-HT3 receptors, which have both 5-HT3A subunits in. So you go to the 5-HT3A gene and you make some of those. How many? Well, it's the less than 5, a number between 0 and 5, but how many I don't know. And then you also make some 5-HT3B proteins and you stick them together. So I don't know how many you take of each, basically. I don't know whether you take 3 of these and 2 of these, or 4 of these and 1 of these. I don't know the stoichiometry. No one does. Okay, so this is what's known as the 5-HT3AB, not Homo Pentamer, oh dear, Hetero Pentamer, meaning that it has different protein subunits making up the pentamer. Okay, so all we know is that there are these 2 receptors that are physiologically important. The other 3 genes have been found, and we suspect, I don't know how much evidence there is for this, but there is growing evidence that you also have 5-HT3A, 3C heteropentamers. In fact, what it appears is that you can either make 5-HT3A homopentamers, or you can mix and match the 5-HT3 proteins with one of the other 4. So you can make 5-HT3A, 3B heteropentamers, and those are definitely known. We know those exist. But you can also potentially make 5-HT3A-C heteropentamers, 5-HT3A-D heteropentamers, and 5-HT3A-E heteropentamers. That's the way that if these exist at all, that's the way we think they're going to exist. Okay, right. So all I can say is that these channels, they have some 5-HT3A proteins in and some 5-HT3B proteins in. How many of each, I don't know. Right. So you have these 2 types of 5-HT3 receptors, the homopentamer and the heteropentamer, often just referred to as the 5-HT3A receptor and the 5-HT3AB receptor. And those are the only 2 ligand-gated ion channels to serotonin that are known about. Okay, now, basically, these 2 receptors have different conductances. They are both cation selective, so when they open in response to serotonin binding, they allow sodium and potassium ions to move through them. Okay? Now, their conductances, however, differ. So this homopentamer has a high conductance. So the number of ions it will allow to move through it in a minute is very good, it's very high. Whereas the heteropentamer has a low conductance. Sorry, I've got this the wrong way around. This one has the low conductance, and this one has the high conductance. So the 5-HT3AB has the high conductance. Okay, right. So the homopentamer does not allow many ions to move through it in a minute. The heteropentamer is much better. And what's causing this? Well, basically, it has everything to do with the intracellular domain. So let me draw out another picture. So here's our receptor, our 5-HT3 receptor. Okay, and we know that on the bottom now, you have, let me draw the 5 subunits first so that I can show the intracellular loops coming off each one of them. Okay, at the bottom, we know now that you have this intracellular domain where you have these intracellular loops coming together like this to form this sort of basket-like structure here with these portals out which ions can move out through. Okay, so let me colour these in. So, in purple, these are the intracellular loops. And I'm only showing 3 because the other 2 would be behind it. Okay, but every single one of these subunits will have an intracellular loop. Okay, so what we find is that basically, if you look at a 5-HT3A subunit, on its intracellular loop, it has a lot of arginine amino acid residues. So let me show you the structure of arginine amino acid residues. So if I draw it here, the structure of the arginine amino acid residue. So here's the amino acid group. Here's the alpha carbon with the carboxylic acid group coming off down here. Okay, and the R group of arginine, you have a methylene group, which you have 3 times, and because I'm lazy, I'm just going to put a brackets around it, and then a little 3 at the bottom. That's a useful trick. Then you've got a nitrogen next, then a carbon with an amino group coming off it, and then a double bond to a nitrogen here with a hydrogen. Okay, and this nitrogen with the double bond has a special name. This is known as the guanidino nitrogen. The guanidino nitrogen. Okay, now the guanidino nitrogen has a lone pair of electrons. So the lone pair of electrons is here. And basically, this lone pair of electrons is a center of negative charge. So you have this center of negative charge here. And protons, okay, protons, so hydrogen nuclei, which have a positive charge, can come and sit by this lone pair of electrons, which is this center of negative charge. And that means that the end of this arginine amino acid gets a proton associated with it. It gets protonated, and that gives it a positive charge because the proton had a positive charge, and the proton's now associated with the end of the arginine, so it now gives the arginine a positive charge. So, these intracellular loops of the 5-HT3A receptor subunit have a lot of arginine in. So this is arginine here. And by the way, just a bit of background information, arginine's 3-letter amino acid code is ARG, and its single-letter amino acid code is R, because it couldn't have A because alanine got A. Okay, right. So, why could that explain its low conductance? Well, if you are lining these intracellular loops with positive charge, then what's going to happen? This channel is supposed to be conducting sodium ions into the cell. Are the sodium ions going to like passing through these portals if the portals are lined by positive charge? Absolutely not. So that is believed to, well, at the moment it's just a conjecture, but I'm going to tell you the experiment that they did to show that that's true. At the moment we would conjecture that that may well be why these channels have a lower conductance than the heteropentamers. Basically, if you look at the 5-HT3B subunit, that doesn't have these arginines. So if you take the arginines out, if you make a mutant 5-HT3A where you've taken the arginines out and replaced them with the amino acids that the 5-HT3B receptors subunit had in the equivalent positions of the intracellular loop, then this new 5-HT3A Homo pentamer, which contains these 5-HT3A mutants, this has a high conductance, just like the heteropentamer. Okay, so basically, by removing the arginine residues from these intracellular loops, you remove the positive charged residues that are lining the portal, and that allows positively charged cations to move through more easily, and that increases the conductance through the 5-HT3A receptor. So that's just an example of how important the intracellular loop is as well for controlling the conductance of the cis-loop ligand-gated ion channel. And we believe that this principle would go beyond, would extend beyond just the 5-HT3 receptor, but also onto the other cis-loop ligand-gated ion channels.